Hi friends, welcome back to my channel Only Bio, presenting up the revision of the chapter Excretion and Osmoregulation. Friends, this is the eighth part of this chapter. I hope that you are following up this chapter. Please do check the playlist so that you can get to know about the other chapters as well. Please share it up with your friends and let's study together. Today we are starting up with the structure of the neck of the nephron. In the last lecture we had a talk about the two parts of the Malpighian body, that is Bowman's capsule and the glomerulus. Okay, as you remember, the nephron is made up of two parts, the Malpighian body and the renal tubule, which are connected with each other by a very narrow area that is called as the neck. So let's begin with the parietal wall of the Malpighian body continues into the neck. The wall of the neck is made up of ciliated epithelium. The lumen of the neck is called as the urinary hole. Friends, lumen is nothing but the space inside the neck, and the opposite end of the same is called as the vascular pole because it's in contact with the tuft of the blood vessels, that is glomerulus. So that part is called as the vascular pole. It is not mentioned in our book, so please remember this. The other opposite end is the vascular pole and the space in the neck is the urinary pole. Friends, our textbooks have mentioned about the urinary pole, but they have failed to mention about the vascular pole. So please do keep this in mind. The renal corpuscle has two poles, a vascular pole and a tubular pole, that is nothing but the urinary pole. The arterioles from the renal circulation enter and leave the glomerulus at the vascular pole. The glomerular filtrate leaves the Bowman's capsule at the renal tubule at the urinary pole. Neck leads to the proximal convoluted tubule. So friends, on the right hand side you can see a beautiful picture of that of the nephron. We are going to talk about the structure of the nephron, especially the renal tubule, because as we have already talked about the Malpighian body in the earlier video. So we started up with the neck of the nephron, which continues into the first coiled part of the renal tubule, which is nothing but the proximal convoluted tubule, also called as the PCT. So it is a highly coiled part of the nephron which lies in the renal cortex and is continuous with the capsular space. It is lined by simple cuboidal epithelium with brush border, that is microvilli, and rich in mitochondria, which helps in increasing the area of absorption greatly. See, microvilli are nothing but finger-like projections. So more the number of microvilli, more is the surface area and more is the absorption. The blood brought by the renal artery is filtered by the glomerulus and then passed to the PCT. Due to convolutions, that is coiling, the filtrate flows slowly and remains in the PCT for a longer duration. This ensures the reabsorption of maximum amount of useful molecules. Friends, please remember the process of urine formation occurs in three steps, wherein the first step is ultrafiltration, which occurs in the Malpighian body. Second is selective reabsorption, which occurs in different parts of the renal tubule, but the maximum of the reabsorption occurs in the PCT. To small extent, reabsorption occurs in the loop of Henle and to small extent in the DCT and the CT. Okay, and the third step of urine formation is tubular secretion, which occurs only in the DCT. Friends, we shall be talking about this process in the next videos. Okay, but right now just remember that these are the three steps of urine formation, of which PCT is actively involved in the process of reabsorption. Okay, now PCT, why is it the main site of selective reabsorption? The reason is it is quite a longer tube, it is highly coiled, so it has a larger surface area for reabsorption. Now friends, remember, just imagine कि if the PCT would have been a strong straight tube rather, so what would have happened? The filtrate would have just entered and exited out. So it wouldn't have spent enough time for this process. So the highly convoluted nature of the tubule helps in retaining that filtrate for a longer time, and this in turn helps in maximum reabsorption. It is a site of reabsorption. Maximum reabsorption takes place in the PCT of the nephron. Friends, once again you can see a beautiful picture of that of the nephron. PCT is the region of renal tubule where reabsorption of essential substances like glucose, proteins, amino acids, a major portion of electrolytes and water takes place. The surface area for reabsorption is facilitated by the cuboidal epithelium in them. Reabsorption takes place at the expense of energy. That is, the process is active. You know, active transport and passive transport, right? So, because it's going to utilize energy, we can call it as active transport. PCT selectively secretes ions such as hydrogen, ammonia, and potassium into the filtrate and absorbs bicarbonate from it. Thus, PCT maintains the electrolyte and the acid-base balance of the body, or rather of the body fluids, you can say. The PCT is surrounded by peritubular capillaries. Let's understand the structure of the loop of Henle of the nephron. See, PCT was in the cortex. From there, it enters the medulla, and while it does so, it takes a turn to form a U-shaped structure that is called as the loop of Henle. Friends, from this you have to understand that the loop of Henle is basically located in the medulla region. Let's understand. The loop of Henle is a U-shaped or a hairpin-shaped tube, that is the middle portion that extends from the proximal tubule. It is composed of two limbs or parts, that is descending limb and the ascending limb. It begins in the cortex, receiving filtrate from the proximal convoluted tubule, then extends into the medulla as the descending limb, and then returns to the cortex as the ascending limb to empty into the distal convoluted tubule. The primary role of the loop of Henle is to enable an organism to produce concentrated urine not by increasing the tubular concentration but by rendering the interstitial fluid hypertonic. Very, very important point worth noting. Both limbs are composed of simple squamous epithelium. The cells have few organelles, little to no microvilli, and low secretion abilities. The two limbs work in parallel with the surrounding vasa recta capillaries to adjust the filtrates, salt, that is sodium, chloride, potassium, and water levels. Friends, this is what is meant by the counter current multiplier system. Now let's understand more about descending limb. It is thin-walled. It is lined with simple squamous epithelium. It is permeable to water and less permeable to salts, that is electrolytes, and thus only indirectly contributes to the concentration of the interstitium. Friends, you may be even asked to distinguish between the PCT, DCT, or distinguish between the descending limb and the ascending limb. So do concentrate and focus into what I'm talking about. As the filtrate descends deeper into the hypertonic interstitium of the renal medulla, water flows freely out of the descending limb by osmosis until the tonicity of the filtrate and the interstitium equilibrate. That is, it comes in balance. The hypertonicity of the medulla and therefore concentration of the urine is determined in part by the size of the loops of Henle. Let's understand about the ascending limb. It is thick-walled. It is lined with simple cuboidal epithelium. The ascending limb continues as another highly coiled tubular region, which is called as the distal convoluted tubule, in short called as the DCT. It is impermeable to water, a critical feature of the counter current exchange mechanism employed by the loop. The ascending limb actively pumps sodium out of the filtrate, generating the hypertonic interstitium that drives the countercurrent exchange. In passing through the ascending limb, the filtrate grows hypotonic since it has lost much of its sodium content. This hypotonic filtrate is passed to the DCT in the renal cortex. The descending limb is permeable to water but impermeable to an electrolyte, while the ascending limb is permeable to electrolytes but impermeable to water. Since the electrolytes get reabsorbed at the ascending loop of Henle, the filtrate gets diluted as it moves towards the ascending limb, but reabsorption is limited in this segment. Countercurrent mechanism is a crucial role of the loop of Henle. The loop of Henle is surrounded by capillaries called as vasa recta. Its function is to operate the countercurrent system, a mechanism for osmoregulation. In comparison with PCT, the DCT has a different structure and function. Cells lining the tubule have numerous mitochondria to produce enough energy, that is ATP, for the active transport to take place. The DCT, which is the last part of the nephron, connects and empties its contents into collecting ducts that line the medullary pyramids. The collecting ducts amass contents from multiple nephrons and fuse together as they enter the papillae of the renal medulla. The wall of the DCT consists of simple cuboidal epithelium. The DCT performs tubular secretion, also at times called as augmentation or active secretion, in which the wastes are taken up from surrounding capillaries and secreted into passing urine. The DCT helps in water reabsorption and regulation of pH of body fluids. Much of the ion transport taking place in the DCT is regulated by the endocrine system, that is, in the endocrine glands and the hormones. In the presence of parathyroid hormone, the DCT reabsorbs more calcium and secretes more phosphate. When aldosterone is present, more sodium is reabsorbed and more potassium is secreted. Atrial natriuretic peptide causes the DCT to secrete more sodium. Similar to PCT, the DCT also secretes ions such as hydrogen, potassium, and ammonia into the filtrate, while reabsorbing the bicarbonate from the filtrate. Conditional reabsorption of sodium ions and water takes place in the DCT. Thus, it maintains the pH and sodium potassium level in the blood. Much of the ion transport takes place in the DCT is regulated by the endocrine. In the presence of parathyroid hormone, the DCT reabsorbs more calcium and secretes more phosphate. Very, very important points. The DCT also consists of straight and convoluted segments. Convoluted is a coiled bar. The straight distal tubule, that is the thick ascending limb, not to be confused with the loop of Henle, continues on from the thin ascending limb of the nephron loop at the level between the inner and the outer medulla. The convoluted distal tubule projects into the cortex. Both parts of the DCT are composed of simple cuboidal epithelium similar in morphology to the proximal tubule. A key difference between them is that the epithelium of the distal tubule has less well-developed microvilli. Reabsorption and secretion occurs here, albeit to a lesser degree than in the proximal tubule. By having lots of mitochondria, the straight distal tubules can reabsorb any useful substance or electrolytes and secrete any remaining waste products using active transport. Friends, let's understand the collecting duct system. Again, you can zoom in and see the picture for you to understand it better. The collecting duct system. Each DCT delivers its filtrate to a system of collecting ducts, the first segment of which is the connecting or the collecting tubule. The collecting duct system begins in the renal cortex and extends deep into the medulla. Collecting tubule is a short, straight part of the DCT. Collecting tubule reabsorbs water and secretes protons. Collecting tubule opens into the collecting duct. Collecting tubule is not a part of nephron, rather, it is a part of the kidney. The DCT from several nephrons empties into collecting tubule. Several collecting tubules then unite to form a papillary duct that empties the urine into a minor calyx and then into a major calyx and finally into the renal pelvis. The collecting system of the kidney is a series of tubes that moves the urine from the nephrons into the minor calyces. Several distal convoluted tubules from neighboring nephrons drain into a collecting duct via connecting or collecting tubules. Collecting ducts then travel through the kidney medulla, converging at the apex of each renal pyramid. Here, several ducts merge to form a single large papillary duct, which is also called as the duct of Bellini. Friends, not to be confused with column of Bertini. This is papillary duct, also called as the duct of Bellini, which opens into the minor calyx through the area cribrosa. Now, this is once again a very important point nowhere in our books. It's the area cribrosa to be remembered. Collecting ducts are termed as cortical or medullary depending on which part of the kidney parenchyma that part of the duct is located. They are made of epithelial cells which get progressively taller as the ducts get larger. So first we have cortical collecting ducts, as the term tells you, cortical. So they are present very much in the cortex and they are made up of simple cuboidal epithelium. The second one is medullary collecting ducts, which are made up of simple columnar epithelium. And the third one are the papillary ducts, which are also made up of simple columnar epithelium. Collecting ducts are the last chance site for water and electrolyte reabsorption from the filtrate, further concentrating the urine, particularly under the influence of antidiuretic hormone. That is, in short, we call it as ADH. No more reabsorption takes place past the medullary collecting ducts. Again, a very important point, not in our textbooks. So, as the urine travels down the collecting duct system, it passes by the medullary interstitium, which has a high sodium concentration as a result of the loop of Henle's countercurrent multiplier system. Because it has a different origin during the development of the urinary and the reproductive organs than the rest of the nephron, the collecting duct is sometimes not considered to be as a part of the nephron. Though the collecting duct is normally impermeable to water, it becomes permeable in the presence of antidiuretic hormone. ADH affects the function of aquaporins, resulting in the reabsorption of water molecules as it passes through the collecting duct. Aquaporins are membrane proteins that selectively conduct water molecules while preventing the passage of ions and other solutes. As much as three-quarters of the water from urine can be reabsorbed as it leaves the collecting duct by osmosis. Thus, the levels of ADH determine whether the urine will be concentrated or diluted. An increase in the ADH is an indication of dehydration, while water sufficiency results in a decrease in ADH, allowing for diluted urine. Lower portions of the collecting organ are also permeable to urea, allowing some of it to enter the medulla, thus maintaining its high concentration, which is very important for the nephron. Urine leaves the medullary collecting ducts through the renal papillae, emptying into renal calyces, the renal pelvis, and finally into the urinary bladder via the ureter. Collecting duct is a long straight tube where hydrogen and potassium ions are secreted to maintain the electrolyte balance of the blood. This is also the region where maximum reabsorption of water takes place to produce concentrated urine. Friends, I hope you've liked it. Please subscribe if you're new to the channel and share it with your friends. Keep studying with me. Thank you so much.